My name is John Griffin. I'm a member of the Eurofins Scientific Advisory Board, and today I'm pleased to moderate a Discover and Beyond panel focused on application of data and models to target identification and validation. And I'm pleased also to be joined by 4 experts working in this field.
I'm Andrea Beccari from Dompé Farmaceutici, and I'm the Vice President of the EXSCALATE platform.
I'm Céline Legros, Business Line Leader for Target Validation, redefining, and read at Eurofins Discovery.
I'm Carleton. I'm the Business Line Leader for Eurofins DiscoveryAI.
Hi, I'm Mounir Tarek. I'm the CEO of NEBULA Biotech.
So we're entering an era of biology where we're able to interrogate biological systems at ever greater scale and with ever greater precision. At the same time, computational infrastructures and techniques are coming online that allow us to then to surface insights from this data, hopefully to drive better scientific discovery. So with an eye towards thinking about what these methods and data allow us to do that we couldn't do even a couple of years ago, let's discuss just where this field may be going, what's on the horizon, and what might even be a little bit beyond the horizon. To begin, what are some examples of the kinds of data and the kinds of algorithmic and artificial intelligence approaches that are being used to productively identify and validate new targets?
Yes, in our internal procedure, we will try to use an approach that combine data and modeling in terms of exploiting the potentiality of knowledge graph to combine the knowledge around the disease and make some assumption that then they have to be validated in a phenotypic screen, for example. So what we do internally is we define a model of the disease, and then we design a set of phenotypic screening and try to resemble the disease and the model, try to find additional. In this case, it's not target-driven, but is effect-driven or efficacy-driven on the phenotypic parameters that we want to see in the assay. And then we do the target deconvolution coming back to the target.
Okay, so effect first, phenotype first,
Yes.
and then the deconvolution.
I'm thinking about this phenotypic reduction, if you will, and how to turn that into a target-based. How do you deconvolute that into a target-based approach?
In many ways, it's challenging, of course, but what we do, we combine different techniques. For example, we do drug-seq. This is a technique when you see the gene induction of the drugs based on the concentration from one side, and then we do the knockout of the genes related to the target that we think are fundamental out of the analysis of the path. So we try to understand the molecule, the effect of the molecule in specific pathway that we know that are related to the phenotype and the disease, and then we find the key elements in the pathway, having then the experimental validation on a target-based assay.
And it seems like one must achieve the right balance between the kinds of data and the amount of data with the algorithmic approach in order for it to be successful. What is the balance between our current ability to generate different kinds of data and our ability to actually apply algorithms to utilize it most fully? Do we find it's more like we still feel we're data limited in some circumstances, or do we still feel like we don't yet have really the optimal kinds of algorithmic approaches to make the best and fullest use of the data?
I think that we are in between. So some part of the knowledge is well addressed at the moment, but if you think that maybe the 30% of the proteins are not annotated in a family, so are not part of an ontology in the system, even if they have to belong to a class. So I think that moving to disease modeling, that is the final scope, I think, to be sure that the target we select translate up to the patients, there is still a lot of pieces that are missing. So there are really few examples of translational disease modeling. That means that we do not know the biological events that produce a disease it's quite impossible to treat a disease-modifying agents if we don't know what we have to modify. So this is the great challenge, I think, today in the data. So understand what kind of data and experiment or profiling of population. There are many examples that we see today, especially in the United States, programs that start sequencing 14 million of people and then following for years, like the UK Biobank example, but a completely different scale. So then if you start collecting blood samples of 14 million people for 10 years with the genetic profiling, then the disease modeling will be unlocked in the potentiality. So it's a change of the scale of the system. As you said previously, now we have to think about the generation. So it's not something is above us, no? But you see also in the UK Biobank example and the atlas that are coming out in terms of protein profiling correlation between target and disease and biomarkers. This is the trajectory. The trajectory start working on the population and then profile population to identify and select patients and disease. And require a tremendous effort also from a government point of view. So who is going to control this data is a question. And this is a tremendous question about modeling such amount of data. But here I think the trajectory is there.
Yeah, I was wondering because we know that when we do modeling on target, specific target, we have all the structures, crystal structure, we are able to do dockings or whatever we want. But if we think about all complex model, complex cellular models, organs or human, how do you curate, prioritize the data? And I think it will be very difficult to share data between entities because the models definitely will be always different. So for some on-target activities and ADME parameters that we could imagine sharing and compiling data from diverse CROs or pharma or startup. But when we talk about having mini organs or human, the modelization, how do we navigate through all of this data and that variability? As everybody or any cell is a bit different. So that's it's where for me we need to test quite a lot, but we will be really be able to modelize this organ or the body on its own.
It's a very important question. And for example, in our case, for example, we develop several phenotypic assays because of course anything make a snapshot of something capturing an effect. So first of all, most of the disease are multifactorial. So the problem that is behind searching for the goal target is that you make the assumption and the bias that there is one single event that can revert the pathology. And this is not actually true in most cases. So it's easy to handle, it's easy to model, it's easy to design molecule, but not fit very well with the patient. So you're completely right. I mean, we have to find a way to handle the change of the complexity. So in some way we have to do target deconvolution. So at some point we have to enter again in the trajectory of what we are very good at, the design molecule on target. The point is not focus at the very beginning with having a solution that is not validated translation. So combine more open on the effect and see it on a higher level, and then find a way to finalize, because if you enter a lead optimization, you need a target. If you want to address side effect, at some point you have to know what you are moving. But I think that today, having also an idea of the global effect and polypharmacology of the molecule, that are just there. I mean, we work with kinases and they say we don't want polypharmacology, but kinases are so important target that by definition you can need 10 to 100 kinases marketed drugs. So the complexity is there. The point is how we handle it. We control the complexity or the complexity control us. That's the point.
Yeah, but at least if you generated phenotypic models, you can imagine using this model to find new targets because you know better the pathway and how the cells and potentially the organs will behave. So that you could, in theory, use this phenotypic model to validate new targets or at least having more ideas and new ideas.
Yes, in our single case, doing the analysis on the pathway induced by the genetic induction on the phenotypic assay, we are able to find 2 pathways that are never reported before to be relevant in the phenomena that we are measuring. Now, coming back, because then a pathway means maybe 10 to 20, 50 targets. So the point is how we can arrive there. But then you have the modeling part, the simulation part, again, support giving you idea. So out of 50 possible targets, if you do simulation and docking and prediction, you can address maybe 10 and 10 can test and validate. Is what we are doing. So we want to identify a panel of potential targets based on the things that we are moving at the phenotypic level to arrive as close as possible to the target of a measure effector of that pathway. I don't know if we will be successful in the identification of the specific target that drive the effect that we see, but I think there are ways well established to go there. Still, you have not be scared by the complexity of the